We are covering the article, The Density of Perineuronal Nets Increases with Age in the Inferior Colliculus in the Fisher-Brown Norway Rat by Maffei et al. This study is very new. It was published in February, so I thought it would be interesting to the class, and many of you may remember that we also learned about perineuronal nets in another article presentation. Some key terms for this article are first perineuronal nets, or PNs. These are lattice-like structures that surround neurons, and we will go more into detail about PNs in a later slide. Glutamate decarboxylase, or GAD, is an enzyme that converts glutamate, which is an excitatory neurotransmitter, into GABA, an inhibitory neurotransmitter. When we talk about GAD+, this implies the presence of inhibitory neurotransmitter, and when we talk about GAD-, this implies the presence of excitatory neurotransmitter. Some background information to address why we are studying PNs in the aging inferior colliculus is that PNs are found throughout the auditory system, but the effects of aging on PNs have never been studied in the IC. So the study is the first of its kind. The study is of interest to us as future audiologists and student clinicians because we often see elderly patients and older adults who struggle with hearing in the presence of background noise. And we know that the IC is a processing center in our auditory system that is robust against background noise. And it helps us with pitch discrimination, intensity coding, and amplitude modulation to help us encode signals like speech. So we definitely want to know what's going on in there. This figure here shows us what a cross-section of the IC looks like. And we learned in Dr. Jennings' lecture earlier in the semester that the central nucleus or core region of the IC is exclusively auditory and dominated by the afferent pathway. So we will focus our attention on this subdivision of the IC, even though the article also examined PNs in the dorsal and lateral cortex regions of the IC. So perineuronal nets, what are they and what do they do? As mentioned previously, PNs are lattice-like structures that surround neurons. They make up about 10 to 20% of the brain volume, and they can be likened to rows that solidify or contain connections between neurons. So because of this, they are critical for the mechanical stabilization of synaptic contacts and the inhibition of structural plasticity, among other things. So now we get to the research question that the authors of this article wanted to investigate, and that is, as the FBN rat ages, do PNs change, and are these changes specific to excitatory or inhibitory neurons? To investigate this question, 15 male FBN rats were selected as subjects and were divided into 3 groups based on age. So we have a 2 to 7 month old group, a 19 to 24 month old group, and then a 28 to 29 month old group. So the rats were anesthetized and immunostaining of the IC was performed to help us identify the PNs and the GAD- and GAD+ cells in the central nucleus of the IC. So this figure here shows us the PNs in magenta, the GAD+ cells in blue, and then the GAD- cells in green. And we can see the difference between these structures in different age groups. So this row here shows us the difference, or shows us the PNs and GAD+ and - cells in the central nucleus of the IC in the 2 to 7 month old group. This row here shows us those structures in the 19 to 24 month old group. And then this row shows us those structures in the 28 to 29 month old group. And just by observing this image, we can see that the PNs in the group with the oldest rats are thicker, We can also see that the presence or expression of GAD plus cells has decreased, and the presence or expression of the GAD minus cells has increased. And to refresh our memory, the GAD minus cells essentially represent the presence of excitatory neurotransmitter, and the GAD plus cells represent the presence of inhibitory neurotransmitter. So to summarize, the expression of inhibition has decreased and excitation has increased. And we know that we need inhibition to maintain temporal precision in our IC, so we're already seeing a bit of a problem. And these arrows here, they represent PN surrounding GAD plus cells, and the arrowheads that we see here, 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 they represent PN surrounding the GAD minus cells. So what are our findings? This graph is a visual representation of our findings. These gray boxes represent the range of density of PNs in each age group. So this gray box here, it represents the range of density of the PNs in the 2 to 7 month old group, this one in the 19 to 24 month old group, and then this one in the 28 to 29 month old group. And as we can see, the density of PNs in the 28 to 29 month old group is the greatest. Another finding was that PNs surround both GAD plus and GAD minus cells, but they more commonly surround GAD minus cells at all ages and are often thicker here. So why do we care that the density of the PNs in the IC increase with age? Well, we know that the elderly and older adult populations often experience difficulty hearing in the presence of background noise, and we learned in last week's lecture on aging about the age-related changes of GABA, or age-related loss of GABA, the inhibitory neurotransmitter. We also learned about the age-related loss of precise temporal processing abilities in Dr. Jennings' lecture on the IC earlier in the semester, specifically when we talked about gap detection in the aging mice. So these 2 points go hand in hand, and we know that temporal precision in the aged IC is often disrupted with the age-related loss of GABA. So the study showed us an age-related increase of PN density, and we know that increase of PN density is likely creating a less plastic environment for the circuits they surround. So this age-related loss of GABA and the loss of precise temporal processing reduces our ability to hear target sounds in the presence of background noise, and this increase of PN density isn't helping because it doesn't allow for our neurons to adapt to changes brought about by decreases in GABA or any other changes to the peripheral and central processing centers. So our next question might be, why does the density of the PNs in the aging IC increase? Well, the authors state that we need further investigation to determine why this happens, but they did brainstorm some ideas. And one reason they brainstormed might be that the changes we see in the PNs may serve to stabilize and protect existing circuits that have maintained their temporal precision throughout aging. Another reason we see a change in PNs might be because our auditory system is attempting to maintain a balance between inhibition and excitation, and then if we remember that the PNs most often surrounded the GAD minus cells and were often thicker here. But again, as the author said, we need further investigation to determine why that is. So that is the end of my presentation, and thank you for watching.